So I'm going to talk about the lung cancer early detection trial that we're involved in, headed up by Tony Ying and Theta Wynn, and we're investigating exosomal markers and immune markers and imaging in this trial. So it's kind of a discovery trial to evaluate novel forms of radiomics for CT for lung detection, lung cancer detection. We're going to evaluate also exosomal and immune biomarkers in the blood, and we're going to form and validate multimodal risk signatures from the imaging, from the blood, or in combination. So we're recruiting patients into Lister Hospital, where Theta Wynn is heading that activity, expecting to recruit 800 suspected lung cancer patients by this month. We've been recruiting for the last 2 years, and we're going to follow up for 2 more. In April, we had 558 patients currently on the book, so it's looking fairly good in terms of numbers. And they will receive then their standard of care, CT scan, diagnosis, follow up with histology, and we also have a blood sample at that time. So what are we measuring? Well, Balaji Ganeshan and his team are doing TexRad radiomics, which is kind of traditional. It's a commercial system now. Traditional measurements of uniformity and granularity in a segmented lesion. So it's doing multi-scale image processing for image texture, and quite a few measurements going forward to outcome prediction and multivariate analysis. Sorry about that, that's to be skipped. Then at King's College London, we've been working with Julia Schnabel's group, and in particular Sam Ellis, who has been developing an autoencoder-based deep learning approach. So the advantages of using this autoencoder is that you can do unsupervised transfer learning from a public data set, and in this case we're using the Luna 16 data set, which has many more samples than we have, and it generates a low-dimensional latent code. And we can use the encoder with new data to generate a latent code that we can also take forward to see whether it predicts outcome or whether it can feature in the multivariate analysis. The other advantage is that it's in some way interpretable, because you can use the decoder part and tweak the latent code and see what happens to the image at the decoder end and see whether different parts of the latent code correspond to speculation or other things that you can see in the image. Also at King's, in Tony Ying's group, we're analyzing the blood. We have blood-based exosomal proteins by Jose and Jolan. So exosomes are very small bodies, extravesicular bodies emitted into the blood by tumor cells. They're very small. We can isolate those from the blood, and then we're doing protein dot plots for specific markers of interest like P4HA1, the hypoxia marker, PD-L1, and other immune markers, and other markers like CD73. Also at King's, from the blood, we're doing immune profile. So this is flow cytometry after PBMC extraction. We have 2 panels for T cells and B cells. Well, T cells and NK panel, and B cell and myeloid panel. And you can see all the different cell types that we're isolating with flow cytometry. Sorry, I have to speed through because I have to get through in 4 minutes. But also we're doing the machine learning approaches and risk signature at King's College. And for that, we're using a Bayesian regularized Cox model. So it's kind of similar to a lasso with a Bayesian hyperparameter, but doing reverse covariate elimination to reduce down this large covariate set. And in the preliminary result, we had 58 covariates. We reduced them down to an optimal signature. And the signature we got at that time, this was about a year ago, we had 3 covariates and a nice area under the curve and a nice prediction of cancer. Hopefully this will improve when we bring in more patients and more covariates in our final analysis. So hopefully I haven't gone too much over time, and it just remains to acknowledge everyone who's on the team and the funders of the trial. Thank you very much.
Hello everyone, can you see my screen? Yes, we can.
That's great. So hi, my name is Rami Mustapha. I'm a researcher at King's College London, working in Tony Inn's lab. I am an immunologist by training, and it's actually my pleasure to give this rather quick 4-minute talk on the HERD study, which stands for Head and Neck Early Relapse Detection Study. Now, very briefly, head and neck cancer is bad. It's diagnosed at a locally advanced stage, and half of the patients undergoing radical therapy will relapse within 2 years. Interestingly, 80% of that relapse occurs loco-regionally, and if detected early enough, salvage surgery offers the best improvement in outcome survival. HERD aims at addressing a clinically unmet need to develop a surveillance pathway to enhance detection of early relapse. Now, as cancer is a multifactorial disease, we plan on using multimodal approaches that I will mention later on to generate a risk stratification model. We hope that using this model, we can identify patients with high risk who could in the future benefit from either more intensive monitoring for recurrence or even from early changes in their treatment modality, such as the addition of immunotherapeutic. To achieve that aim, the study will recruit 200 patients classified as intermediate or high risk who will undergo radical treatment as part of their standard of care, as this chart indicates. Now, these patients will undergo imaging as well as multiple sampling, and I think I need to pause briefly on this slide to explain. So imaging will happen either pre-treatment or after surgery. Tumoral tissue samples will either be from biopsy or from surgical resections, and we're also collecting blood and saliva at multiple time points. So if you're wondering what we're doing with these samples, the simple answer is we're sort of doing everything. I tried, but I decided it's impossible to present everything in the 4 minutes I have for this talk. So briefly, we came up with 3 hypotheses for this study, and in order to answer these hypotheses, we decided that we need to do everything from genetic sequencing to cell-free DNA to exosomal biomarkers such as hypoxia. We're looking at the immune landscape both at the tumoral level as well as circulating in the form of PBMCs, pretty much what Paul showed before. Heck, we're even generating patient-specific organoids. We're culturing these organoids with microbes, which we've gotten from the saliva to replicate the microbiome, and we're treating them with standard of care. But since this is an NCITA meeting, I thought I'd focus on the patient imaging aspect of it. And in the case of this study, the research imaging aspect is magnetic resonance elastography. Now, for those of you who don't know, very briefly, MRE entails the generation of mechanical waves through the tissue of interest to scan. Now we use a specific sequence to image changes in this wave as it moves through the tissue. And using some mathematics, this gives us mechanobiological properties of the tissue, in this case of a tumor, which are usually invisible to a standard scan. Now those aspects are like tissue stiffness and tissue viscosity. Professor Alcinkes, who is a co-investigator on the study, has a patient MRE setup running at Guy's Hospital for a breast cancer MRE clinical trial. Now, unfortunately, since HERD is delayed and we have not started recruiting, I thought the best way of explaining the potential of MRE is by showing some data from that study. The aim of the study was to non-invasively detect metastasis in breast cancer, and indeed, you can see that using tumoral pressure measured by MRE, they correlated that with lymphatic invasion in the study. Furthermore, we have also used MRE, so magnetic resonance elastography preclinically, where we had 2 models of head and neck cancer with various resistance to chemotherapy. Now we wanted to see if after 1 dose of cisplatin, so this would be the equivalent of very early on in the tumor, in the patient treatment pathway, we can see changes in the tumor using MRE by assessing something called phase angle. Now the phase angle is nothing more than a correlation between tissue viscosity and tissue stiffness. So we gave these mice 1 dose of cisplatin, and even before we observed any changes in the size of the tumor at scan, we noticed that the resistant model had decrease in its phase angle, whereas the sensitive model had an increase. We moved on to correlate this with changes in the extracellular matrix, which had some interesting implications, but focusing on the patient aspect of this, using the breast cancer trial, Ralph actually had access to patient scans after 1 cycle of neoadjuvant chemotherapy. Now, normally these patients would be assessed after multiple cycles of NAT, but what would happen if we applied the same methodology from the preclinical study? And indeed, we did that, and what you're looking at is a figure showing the relative changes in phase angles. So everything higher than 1 means that the phase angle at the time after the first dose at that scan relative to the baseline scan had an increase in their phase angle, and you can see that actually all these patients were either the complete responders or the partial responders, whereas everything lower than 1 meant a decrease in phase angle, and the 4 non-responders of the study had a decrease in their phase angle. Now, this was very interesting as we actually managed to replicate preclinical data. I think that I decided to stop it here because there's a lot of aspects of the study. So moving on to the acknowledgements, I would like to thank, actually, I do apologize, this is missing the funders for some reason. The slide was complete before. I would like to thank everyone involved in the study, especially the NCITA CTU who are setting this up. And thank you for the organizers. I hope I did not exceed my time.
Hi, good afternoon. Thank you for inviting me. So my name is Giorgio Brembilla. I'm a radiologist and clinical research fellow for the UCL Center for Medical Imaging. And I'm going to talk about the LIMIT prostate cancer study, a study on prostate cancer screening using novel MRI techniques. So as already said, prostate cancer is the most common cancer in men with excellent survival rate when for early stage detection. So it is known that PSA-based screening may have reduced prostate cancer mortality, but also they exposed to overdiagnosis and overtreatment of indolent tumors that would not cause harm to the patient. So there's still room for improvement. And one tool that we could use is imaging, as for other screenings. For example, multiparametric MRI allowed to reduce the detection of indolent disease and to avoid unnecessary biopsies in up to 30% of men in a diagnostic setting. The main limitation of multiparametric MRI for screening are long scan times, limited accessibility, and high costs. It requires gadolinium-based contrast media injection, and also it has limited specificity, so still 1 in 2 men undergo unnecessary biopsy. And which are the potential solution to address these issues? Well, they can be ultra-fast MRI protocols or novel MRI techniques such as Luminal index MRI. Abbreviated bioparametric MRI, it's an ultra-fast MRI protocol that requires less than 10 minutes to be performed without use of contrast media. So it could increase the acceptability, the accessibility, and it could reduce the cost of MRI. It has similar diagnostic performance to a full multiparametric MRI protocol. And as already said, from our imaging screening study preliminary data, we have demonstrated the effectiveness of the recruitment strategy for prostate cancer screening with MRI via primary care approach, and that MRI has a value in early detection of prostate cancer. And also the Prostagram study used the MRI for screening in prostate cancer and was associated with more men diagnosed with clinically significant prostate cancer compared to the PSA alone. Luminal index MRI, it's a novel MRI technique that requires less than 10 minutes to be performed without contrast media use. It assesses biological property of the prostate, namely the prostatic glandular lumen that is reduced in clinically significant prostate cancer. So it has biological specificity. And in fact, from our preliminary data, we observed that luminal index MRI may have higher specificity compared to multiparametric MRI, and so it's expected to further reduce false positive results and unnecessary biopsies. So the LIMIT prostate cancer studies aim to assess the feasibility, the accessibility, and the performance of prostate cancer screening using not only PSA density in this case, but also luminal index MRI and abbreviated bioparametric MRI in men aged 50 to 75 years without prior PCA and prostate cancer diagnosis, with no MRI contraindication and able to consent. It will be, because it hasn't started yet, it will be a multi-site true recruitment strategy study. The further recruitment strategy will be as for reimaging study already done via GP practices, and the alternative, so the second recruitment strategy will be via community approach. So participant will be approached from a variety of sources, for example, radio station, social media platforms, places of worships, barbershops, and so on, because the aim is to recruit, for example, men among the Black ethnicity that, as we know from current screening program and from imaging study, are underrepresented in a screening program. A single study visit will be required in which patient will undergo multiple MRI tests and the blood tests. And in the community approach, the MRI will be, the MRI and the blood tests will be performed on a mobile MRI scanning van that will be located in places of interest throughout London and Manchester. And tests will be provided on the day. As I said, the intervention will be luminal index MRI, abbreviated by parametric MRI and blood test, and men who will test positive for at least one of the screening tests will be referred through the NHS referral pathway for suspected prostate cancer. Which are the outcomes? Well, the primary outcome will be to determine the prevalence of men with at least one test result positive, requiring a referral for suspicious of prostate cancer. That is the screening rate of the screening test. Secondary outcomes will be to assess the uptake and acceptability of screening invitation and to evaluate the diagnostic yield and the net benefits of the screening test. There are tertiary outcomes that will be to derive AI tools for automated reporting and also translational study on blood samples. So LIMIT could be a study to inform on the feasibility and performance of an MRI-based prostate cancer screening strategy using novel MRI techniques. Thank you very much for your attention.